Thank you very much for the very nice talk. The floor is open. Xavier, would you like to talk? Ask questions?
Thank you, Peter. Thank you for these results, Elizabeth. Very interesting. I was wondering if your model, if your cellular in vitro model was sensitive enough to detect sometimes the possible pathogenicity of COL3A1 variant, what we call variants of unknown significance. We have the problem with patients, as it was said before, for one at least of them, with non-glycine missense mutant, but rather something like proline variants or histidine variants, where we don't know exactly if these are functional or not. Did you have the chance to test these? And if it was the case, was it abnormal in your model?
So these are the only cell lines that we've looked at, and they were all confirmed mutations based on, I think we acquired them from the Correll Institute, and they're commercially available. So we haven't assessed what you're talking about in our model, but I think if we had a sample of the cells, I don't see any reason why we couldn't characterize this in the same way.
Thank you.
Do you think that things would look different if you had smooth muscle cells to culture? And could you look at effects that you might think would have a vascular wall effect? I mean, there are very certainly significant skin phenotypes in people, but the money is in the vascular wall at this point.
Yes. Yes, you definitely can produce cell-derived matrix using the smooth muscle cells, and I would be really interested to see if there were changes from these results if we had smooth muscle cells available from patients. I've thought of regressing these fibroblasts back to iPS cells and then redifferentiating, but we haven't gotten there quite yet, but I think that would be a nice next step. We're also attempting to look at the impact of these cell-derived matrices that we have here on vascular cell phenotype. So one of our next steps is to eventually plate endothelial cells or smooth muscle cells on these matrices and see if there are differences in cellular response.
Peter, I had a related question. So the relationship that a thinner matrix is stiffer doesn't seem intuitive to me. I was wondering if you've looked at other matrix elements that may be expressed in a compensatory manner. So if you, for example, look specifically at type 1 rather than type 3 collagen or other matrix elements, might you see something that was responsible for these changes in matrix performance?
Yes, I am ramping up to look at the content of these matrices overall. I've talked with one of our cores that we have on campus about doing proteomics on our matrices to look at the content overall and see if there are changes. We have looked at overall glycosaminoglycan content, and we've seen that it was increased in all of the VEDS cases. So it doesn't really give us an answer as to why the stiffness is increased here, but we're beginning to look into compositional changes.
You might also look at the RNA profiles from the cells before you know, without taking them away, just to see what they're expressing. That might give you an early peek at what's going on in the matrix.
And Xavier asked you a related question, which is whether you've looked at type 1 collagen variants from patients, say, with osteogenesis imperfecta to see if you get similar sorts of alterations.
We have not, but that would be really interesting. I'd be interested to look at that.
Thank you very much for that very interesting talk. Lisbeth Siderius.
Yes, in Europe, there's much action on interoperability of digital health or digital health. One way is to document everything in international standards like the International Code of Disease and SNOMED. Are these also familiar to you? And how else do you search records if you need some kind of query definition?
So because it was 18 patients that we identified, I did the review of the record myself. So I went through doctor's notes, problem list, and the imaging history that was all available in the electronic health record.
Oh. Do you think these international classifications could help?
I think if you have a lot of patients, it would be really difficult to go through each one individually and go through all their past medical notes like I did. So I think using ICD codes could be really helpful in a larger cohort.
Okay, thank you.
One quick comment about that. We tried to do that with some local data and ICD-9s for VEDS, and it's really, a lot of the codes that we want to use are really non-specific in ICD-9. I mean, yes, on huge cohorts, you sometimes are forced to use it, but a lot of the outcomes for VEDS have really vague ICD-9 and 10 codes.
Al?
I may have missed this, but were family members recontacted, evaluated clinically and genotyped?
No, so we did not look for family members of the probands that we identified genetically. We just looked at the people. That turned up with COL3A1 mutations in the biobank, and then looked at their electronic health records.
Is that permitted according to your consent to recontact and do dedicated analyses?
It would be permitted, but yes, we haven't looked into that yet.
Did you confirm the mutations by a second run, or did you rely on the sequence? And how deep was the sequencing depth?
We just relied on the first run. Yeah. The depth is about 30x as well.
30x, okay. And they were all clearly heterozygous at that depth?
Were they all clearly heterozygous at that depth?
Right, yeah.
Yes.
Yeah, Bruce is I don't know whether Bruce is sitting next to you, but he's somewhere in there.
He is.
Are you asking about possible mosaicism, Peter?
Well, I remember talking about this topic and talking about the frequency of apparently pathogenic variants at the Marfan meeting on the basis of the NOMAD sequences, and was told unequivocally that that many of them were found there. The one-offs were errors, but so I was just asking about that. I think mosaicism would be an unlikely source, but would be a possibility. The other is, what's the range of, you said that you had one aspartic acid and one valine substitution and a deleterious splice site mutation, but were the other ones much more predicted to be mild?
Do you mean like in ClinVar, or do you mean?
Just the substitutions, small amino acid substitutions for glycine as opposed to larger ones.
So we did have one glycine to alanine substitution, and that patient did have varicose. Well, we had 4 total, but one of them had varicose. Oh, it's noted to have varicosities. And then the other substitutions in the patients that had minor criteria was an arginine substitution, and that was also a varicosity.
After I read your abstract, when we first looked at them before, I looked in NOMAD for the variants, and they're about half as frequent as they are in your study. So it suggests that they are out there. The frequency, I think that your point is very much that these people are not being recognized, and that the recognition would lead to a higher frequency. The other is that you are looking at people who are being ascertained in hospitals, so they may not be well, which would be another contribution to higher frequency. even though they're not being recognized as having this condition, and despite your careful review of the papers. And I think it's very interesting, and it gives us pause about the frequency of the condition. Other comments?
Peter, I'll just jump in with one thing.
Yeah.
Are you muted? And that is the likely, you're not. Okay, these are not likely to be mosaic, Peter. We clearly have to check, but we've gone through this already for other disorders, and we have ordered the DNAs and gone through the sequencing, and using the same criteria for calling variants, they've all turned out to be heterozygous.
Mm-hmm.
So that's not proof in the formal sense, of course, so that's fair. But on the other hand, at least experientially, using the criteria we use in the range of allele balance and so on, these are not likely to be mosaic.
Mm-hmm.
So was there a final conclusion about how we should be counseling patients who have COL3A1 predicted deleterious COL3A1 variants found as an incidental finding? I mean, I would think that we would still want to report that to patients that had requested to hear about medical information that was potentially actionable.
Yeah, I think we definitely, I don't think this is, if you have a deleterious variant to report, I think it's important to share that information with patients, especially as you said, if they requested that information. But I think what this study was showing, but again, from a small patient cohort, is that even with these deleterious mutations, their outcomes are okay. They survived their pregnancies, they've had histories of surgeries and invasive procedures.
These are the 126 mice of humans.
Yeah.
Or they're the people, that we know plenty of people with vascular EDS who are perfectly fine until they're not. So it's hard to say what next week would have brought for these folks.
Yeah, average age was pretty, the average age of the living subject was 54. So it is pretty late, not late, but like mid-late.
Right. One of the other places to look at this is actually when probands are ascertained to look at their relatives. And I think both the French studies and the ones that we've done here on larger populations find some difference in phenotypic presentations among the relatives. I mean, but many of them it's hard to tell because they've died prior to the time of ascertainment. Thank you very much, Nora. Are there questions? Shane, you had a number of comments as the talk was going on. Would you like to bring that up?
This is just so interesting because we both presented our pediatric experience, and our cohorts were pretty similar in age and follow-up time, both about 5 years of follow-up. I was really surprised just to see that yours had such a predominance of glycine substitutions, while ours was mostly splice site, and that your cohort had a lot more events. And I just wonder if the way that they're coming to attention is different, or I just didn't know if you had any thoughts that was pretty different between our two groups.
Not really, no.
No.
No, I'm not sure why.
Were you able to go back and look at the kids that, the young boys that presented with really life-threatening events, and to look at each one individually and see whether the diagnosis could have been made on clinical grounds earlier? This comes back to the question of what do we do about educating pediatricians and family medicine people about diagnosis. Was there a history of unexplained bruising? Were there other features that might have led to diagnosis in them?
In this group of patients?
Yeah.
There are some history of easy bruising and nosebleed, and some of them were initially investigated for bleeding disorder, but was then dismissed as nothing. but I can't exactly remember which one in the group. Yeah.
We've been in contact with people who are involved in trying to put together the pediatric guidelines for what to do, and what do you do if you come to the end of of an evaluation for easy bruising in a pediatric setting, and should you proceed on? What are the circumstances under which you would proceed on to doing genetic testing for vascular EDS, for example, or some of the other conditions?
It's so low cost now. You wonder if there's certain things that are rare, like pneumothorax in a child, or that in these bleeding workups that could trigger a workup. I mean, just the cost is less than an echocardiogram.
Right, and certainly if you do a combination of things like clubfoot easy bruising, and spontaneous pneumothorax would all be, you get a trifecta, and you're off to the races to do testing. But you're right, genetic testing is now, or can be now, cheaper than almost any other test that's done.
Right, and a large proportion of our cohort didn't have the easy bruising, so that was what, I mean, I think 60 or 65% did, but still like 35% didn't, so it's just interesting.
So we've all been contacted by coroners to investigate sudden death in young people. And when that diagnosis has turned out to be vascular EDS by molecular testing, it's always been clear in retrospect that that was the right diagnosis, and that diagnosis could have been made if that person had seen someone with familiarity with the condition. So I wonder whether there's a broader role for genetics in general, both molecular and clinical, in the evaluation of kids with unexplained bruising or some of these other EDS features.
Well, we consider medicine a sub-discipline in genetics as opposed to genetics being a sub-discipline of medicine, and so I think that's always the case.
Can you hear me?
Yes.
Hi, it's Elena Cervi. I just wanted to reply to Shane about comments on the difference in our populations. We actually do not have any prevalence of congenital heart disease. So we have this one with pulmonary atresia and interventricular septum, and another one where a fetus was diagnosed in utero with PAIDS, and the pregnancy was terminated because they also had vascular EDS, so the parents decided not to go ahead with the pregnancy. But other than that, we have nothing else at all, not even minor things. In terms of the vascular events in the two boys that died during the adolescence, one of them was a known diagnosis, and they were just lifting furniture, and he had a type A dissection and died of it. The other one was not known, and it was a post-mortem diagnosis with molecular autopsy. So I think in that case, he never presented to medical attention before the event that actually led to his death. The other ones, most of the very young ones, presented with quite dramatic events, and it was spotted by either the pediatrician or the cardiologist looking after them that there was something wrong because the presentation was quite dramatic. But again, the subdural hemorrhages and the splenic rupture were considered non-accidental injury as a first hypothesis, just to highlight the fact that sometimes it's not the first thing that comes to mind.
Right, and it's true, as we brought up before, that the high incidence of congenital heart disease in our population might have to do with our center, that we're a major congenital heart center, so that might have introduced some bias into our high numbers.
In a way, I don't know. I mean, Great Ormond Street is probably the largest center in the UK in terms of congenital heart disease. So our population is very much congenital heart disease based. But how it works in the UK, there's a national diagnostic service that is based in Northwood Park. So most of our referrals come from the genetic assessment in Northwood Park. So we also have those that wouldn't necessarily come to general pediatric cardiology attention for any other reason. They already have a diagnosis of vascular EDS. So that's why probably we see more predictive genetic testing in families that have the disease already known. And again, I think the probands are the ones that I'm more concerned about, because it seems like we have a lot of events and again, quite dramatic.
So I think we have to move on. There, I think you can look through, you'll see comments from Jessica Kushner, others about things. We have a little bit of time for discussion after the next session. But thank you all very much.